The webinar, New Horizons in Plant Breeding, Genome Editing, is about to start. Before we begin, let's quickly review some housekeeping items. Please note that participant phone lines will be muted for the duration of the event. If you need assistance at any time today, please press *0 and an operator will assist you. For technical support with the web portion of today's program, you may send an email to webinars@pma.com or use the chat box in the lower left area of your screen. If you wish to ask a question at any time during this presentation, simply type it into the chat box and then click on the Send button. Now I will turn it over to Bob Whitaker with PMA. Please go ahead, sir.
Thank you. Hello and welcome to the 2017 PMA Science and Technology webinar titled New Horizons in Plant Breeding, Genome Editing. I'm Bob Whitaker, Chief Science and Technology Officer at PMA and your moderator for today's webinar. PMA is proud to bring you value through webinars such as this one. Our goal is to provide year-round educational opportunities and connections to those involved in the produce industry. In addition to webinars, you can access more research and technology-specific information on PMA.com and through our other networking and education events. Mindful of the trend to feed an ever-expanding population that is expected to approach 9 billion by 2050, and to do so with less human and natural resources, one area of technology that we've continued to focus on in these webinars and throughout PMA education and outreach is plant breeding. It is important to leverage the genetic capacity of plants and specific genetic tools to develop new varieties of fruits and vegetables that can be grown more sustainably, provide better nutritional profiles, deliver an improved eating experience, and have increased yields. In the last few years, the tools to permit genome editing have garnered a great deal of scientific and media attention. The ability to precisely edit or make changes to a plant's genome to enhance a desirable trait or perhaps negate an undesirable one opens up exciting new possibilities for the creation of the next generation of fruit and vegetable varieties. Therefore, this webinar today will provide us an overview of emerging work on advanced breeding and specifically the CRISPR-Cas genomic editing system and how this technology is currently being applied and what we might expect in the future. We are indeed very fortunate to have Neil Gutterson, Vice President of Research and Development for DuPont Pioneer, as our speaker today. Neil leads R&D and product development functions to create innovative agricultural products, services, and integrated solutions which combine Pioneer's industry-leading germplasm, breeding systems, biotechnology-derived solutions, and data analytics. Neil has built his career through a series of progressively more senior research and development roles at biotechnology startup companies, including Advanced Genetic Sciences and DNA Plant Technology Corporation, which is actually where Neil and I worked together for a number of years. In 2002, he joined Mendel Biotechnology as Vice President, Research and Development. Neil has also served as President, Chief Executive Officer, and a board member at Mendel Biotechnology prior to joining DuPont Pioneer in 2014. Neil holds a Bachelor of Science in Chemistry from Yale and a doctorate in biochemistry from the University of California at Berkeley. He has also attended the Stanford Business School Emerging Companies Executive Program. Neil is the inventor of more than 30 patents and patent-pending applications. I think it's safe to say by all that Neil has been a pretty busy guy throughout his career of extreme accomplishment. Before I turn the webinar over to him, I would remind you that if at any point during the webinar you have a question, simply type it into that chat box area and then click on the Send button. We'll do our best to address your questions in the Q&A session. So without further delay, I'll turn the webinar over to Dr. Neil Gutterson.
Well, thanks, Bob, for that lovely introduction, and thanks to the PMA for inviting me to participate in your webinar today. So for the next 30 or 35 minutes, what I will do is interweave some perspectives from the DuPont Pioneer viewpoint about the technology and how it'll be used and how we'll use it, with some pretty basic framing of the technology itself, what it means, what it is a little bit, and how one might think about using it more broadly. So on this slide now, what I want to talk about for a moment is just the challenges that I think we all face in serving a growing population with healthier food. And we know at DuPont Pioneer that the ultimate end user of what we're doing is the consumer. And what the consumer wants today is healthier food produced in healthier ways that helps make them healthier. Our goal at DuPont Pioneer is to enable our farmers, our customers, to serve that need of the end user, of our consumers. And we know that the key issues for our customers today are still grounded in fundamental productivity, improving crop productivity. And that means a number of different issues about their production systems. Ultimately, we deliver improved genetics. They count on us for that improved genetics. And that improved genetics delivers increased annual yields. It delivers better performance of the final products. And they count on us to help them with protection against pests and pathogens so they can realize the ultimate value of what they plant on their farms. Now, to do that, to serve our farmers, we in research at a company like Pioneer have a series of platforms that we use to provide solutions for them. And you can see on this first slide the series of platforms that we have today. And it's a diverse set. So this is within the context not only of Pioneer as a seed company, but of DuPont and our overall ag business. And so it includes breeding as our most fundamental anchor of value creation. But you can see as the second on that list, circled here, is CRISPR as a way of really pointing to genome editing as a new technology that will feed into our breeding programs to bring new value. And of course, there are a number of other platforms that we have for biotech traits and crop protection solutions, et cetera. What I'll spend most time, of course, talking about is CRISPR and explaining its uses. So before I get into the actual technology itself, I just want to acknowledge what perhaps all of us are aware of, which is, as Bob noted, the level of excitement and interest, both amongst academics and amongst the public at large. And so I'll start on the right side and just point you to a few examples of publications over the past couple of years that highlight a range of applications of CRISPR-Cas-based technology. Everything from, as you can see on the left particularly, the idea somehow of editing humanity. And there's a lot of enthusiasm, I would say, not so much for editing humanity and changing the germline, where we have a lot of caution, but in fact there's a lot of enthusiasm for medical treatments, for therapeutics that could address important problems, blood disorders or cancer. And then all the way over to solving problems that we're talking about, hunger and pollution potentially. So on the left side, you can see the explosion in interest in the academic community. And if you go back into the early 2000s, there were very few publications around CRISPR-Cas as a natural phenomenon discovered in bacteria as part of the immune system of bacteria. But once the publication in 2012 was made about the application of this natural system to editing any genome at all, you can see a rapid expansion in the rate of publications in the academic community. And this is not tapered. This is still growing today. So we have been interested for a very long time at Pioneer in finding ways to edit the genome in targeted ways. And if you look at this slide, you can see some of the different technologies we've tried over the years. And it goes back to, you can see the names, zinc fingers and meganucleases and talons, and highlighted in orange at the top right is CRISPR-Cas with the discovery in 2012. And this is our view, having tested a number of these technologies, of the efficiency and time perspective, the quality of the outcomes, the cost efficiency, and the technical flexibility of the different tools. And what I can tell you is that CRISPR-Cas is a step change in utility compared to any of the other earlier technologies. And that is why you may not have heard much about genome editing in the 2000s, but in this decade we're hearing a tremendous amount about genome editing as a tool for a wide range of biological applications. And so to give you a sense of why this has happened, if you think about the three other examples of technologies, zinc fingers and meganucleases and talons, those are all protein-based systems. And I'll get into explaining a little bit more about CRISPR-Cas in a moment, but in order to address a particular location in the genome, because these are protein-based systems, every time a new address or target in the genome has to be found, for those earlier systems, a new protein had to be designed. And that's rather complex and tedious work. On the other hand, CRISPR-Cas is itself a protein that does the job of initiating editing. But to target locations of the genome, all we have to do is append to that protein a very simple nucleic acid, which we can synthesize purely based on the target sequence in the genome. And so I'd offer you an analogy here to think about, which is that for all those technologies that are protein-based, imagine if every time you needed to make a phone call, you had to go and buy a new cell phone. You wouldn't make a lot of phone calls. But if your single smartphone was adequate just to type in, as we do today, any number, then you make lots of phone calls. And that's where we are today. That's the relative ease of CRISPR-Cas compared to the early technologies. That's why there's such an explosion of interest today. So when we think about this as a tool, I want to anchor you to the concept that this is fundamentally about breeding and builds on the lineage of a company like ours of over 90 years of commitment to bringing advanced genetics to our customers, starting with the work of Henry Wallace, bringing the first hybrid corn in 1926, developing the first hybrid called Copper Cross. And this slide shows a number of improvements over the years, both in breeding as a foundational tool, and the innovation of Henry Wallace was about hybrid systems, through to the next sort of big step change in crop improvement of ag biotech in the 90s, with its applications for insect control and herbicide tolerance in particular, but some other examples. And on the bottom you can see an example of Plenish, a high oleic soybean that we've developed and is in the market today. And then up through what we think of as what will be the next really big third revolutionary change in crop improvement. And as you can see from these arrows, these all build on one another. We don't stop doing hybrid systems because we have agribiotech. We don't stop doing breeding and germplasm improvement because we have CRISPR-Cas advanced breeding. These all build on one another as we bring new products with new technologies to the market and recognize and take advantage of the importance and power of these technologies. So how can we use CRISPR-Cas? The next slide takes us there. And the analogy I want to give you here is to a search and replace system that we're all familiar with as on your word processor on your computer. So you may have a large text. A typical text maybe is a couple of 100,000 characters. When we're thinking about genomes, the texts are much larger. Typical corn has maybe 3 billion characters, and that's similar to the size of the human genome, 3 billion characters. But nonetheless, we have to have a system that will take us to a location, search in the genome, and then be able to introduce an edit. So again, a search and replace function as in text editing. And so the three things we can do with CRISPR-Cas is to delete a portion of the text, to edit a portion of that text, or to, in a very particular way, search to a location and then replace one word, one part of the text with another. And so we can deliver a wide range of really desirable outcomes with those three types of changes. And on this slide, you just get examples of those. So, there may be a text in the genome that essentially is repressing how to produce a particular output. You can just delete that part that says not, and you can deliver something that has the desired output. We can edit in more subtle ways. So the text may say something about a gene, for example, for tolerance to drought, and we can make a subtle change and edit and get high tolerance to drought. And finally, we know there are locations in the genome where there may be a gene that gives some level, perhaps, of resistance to disease, but basically not very much. And in another genome of the same organism, let's take corn as an example, there is a gene that's much more effective, giving resistance, and we can actually replace that more susceptible allele with a resistant allele. And that's another really powerful opportunity and application of CRISPR-Cas-based targeted breeding. So let's step back for a minute and just talk about the technology itself and how it works, and really how it relates to very natural processes that we're pretty familiar with. So on this slide, I'm illustrating that any crop in the field is undergoing stresses of various types. Heat stress that might lead to oxidative damage, radiation from light. And as a result of those stresses, double-strand breaks occur in the DNA in the genome. Now, because that happens so frequently, any organism, all organisms, have developed repair mechanisms that can heal that double-stranded break, and as you can see in the DNA repair on the right, restore that to its normal state. However, in that DNA repair process, it is often occurs that we get some small change, either a deletion of one or a few bases, or even the addition of one or a few bases. As a result of that, one ends up with a mutation. And so in nature, occurring all the time is this process of double-stranded breaks and repairs that can lead to perfect healing or to mutational repair that leads to changes in gene function. So we can harvest that. We can harvest that by using the system I described with the CRISPR protein being able to bind to an RNA molecule that targets a particular location and therefore delivers a double-stranded break directly to a location in the genome that we have in mind to create either a deletion, an edit, or to replace a particular gene at that location. So what does that mean to us? How do we use that in conventional breeding? And why is it powerful? So this slide illustrates an example that we can easily conceive of in corn, where we would have a variety, let's say at the bottom, that is sensitive to drought, but it's a really high-quality variety. Let's say it's adapted to Iowa, where I'm sitting today in Johnston. But drought's a problem, and we want to improve the drought tolerance of that variety. We know there's another variety that might be a tropical variety that has really good tolerance to drought, but it doesn't adapt very well to Iowa, and it's genetically pretty diverse. So the way we would take advantage of that variety with its tolerance to drought is by a series of crosses, where at each stage we would reduce by half the number of genes that is coming from that tolerant variety till we get down to just, ideally, one or a very few genes that give us tolerance to drought, and then retain otherwise the higher quality variety that's adapted for the target environment. And so the goal is to get a high-quality plant with drought tolerance. To get to that end goal could take many, many, many cycles of crossing. In contrast, on this slide, on slide 9, you can see what we can achieve with a technology like CRISPR-Cas that enables us to essentially create that desired drought tolerance characteristic directly in the hybrid background. In the case of corn, because it's a hybrid, we would actually introduce it into one of the inbreds or maybe both of the inbreds that we bring together to create the hybrid. But the fundamental point is that in only 1 or 2 steps, instead of perhaps 7 to 10 steps, we can create that same desired goal, and it's actually a higher quality outcome because it's done exactly in the elite varieties, and we make only a single change, and there's no additional genetic linkage of other parts of the genome coming from the drought-tolerant variety. And so you can see the efficiency and the quality that's achieved using a technology like CRISPR-Cas for targeted breeding. Not only do we get a really high-quality outcome, but as you can see in this slide, this can be a very, very efficient process. Now, I'll just pause here for a moment and say that the efficiency of the process of genome editing will depend upon our knowledge of the biology of the target gene and the outcome of modifying that target gene. The good news is there are many targets we know today that we've wanted to be able to address for a long time, and there are many genes whose behavior, once we would maybe delete them or otherwise edit them, we know exactly what to expect. So in those cases, we can see that, and an example of waxy corn, which I will come back to in a moment, you can see that we can deliver an outcome in as few as 5 years. 5 years from the start of the initial edits through to field testing and then advanced testing in large typical environments, multiple environments over different geographies, to then validate the performance and get to the market. In contrast, even for our traditional plant breeding systems, we're talking about an 8-year process from the start of the breeding cycle to a commercial planting. And even more dramatically, if you were focusing on a transgenic trait to deliver a desired outcome, we're looking at at least a 13-year process, given all the testing required and regulatory requirements to bring that product through to the market. And so we talked about quality of the product, and you can see here the time efficiency is very tantalizing for all of us who deal with the development of new varieties and their commercialization. The opportunity to bring targeted traits, whether those are for the farmer's benefit or for the end user's benefit in a quality trait, any and all of that can be done in a very cost-effective way. And we think this will be a really important component of our breeding systems going forward. All right, so how do we then use this overall? What are some of the applications that we have in mind? Just to illustrate the breadth of applications, to give you a sense on the phone about the kinds of applications you might think about for the produce that you're handling and selling today. So let me start on the right-hand side and give you a broad picture. So on that table on the right-hand side, you can see a series of crops on the left axis: corn, soy, oilseed rape or canola, rice, wheat, sunflower. These are a series of crops that we are actively commercializing today, and we foresee, as we go out into the next decade, that we'll begin to bring products forward into all of those different crops. And across the top of that chart, you can see some of the categories of traits that we think are both very amenable with this technology and very important to our customers. And really, to be honest, it starts first with what's important to our customers, working back then thinking about how can we use the right technologies, and we see genome editing, targeted breeding as the right sort of technology here. So for example, disease resistance, very important in all our crops, very important to all of our farmer customers around the world. And we expect to be addressing and improving disease-resistant products for corn, soy, canola, rice, wheat, sunflower around the world. Other cases, yield stability. There are particular reasons why we think those particular crops are attractive targets. Drought tolerance, you can see examples in corn and rice where we think this is important as we think forward to the needs of the world and challenges with water availability, whether that's corn in the U.S., or rice in India, for example, where we know water will become more and more scarce. The ability to address drought tolerance, we know, will be accelerated by this technology. Importantly, we also know that there's an opportunity to improve output traits or the quality of the product. And in our case, we're focusing here on 3 oilseed crops where we can modify oil composition, or because they all also then produce protein, or in the case of soy, protein is really the target, we can also modify the protein composition of those products and deliver different protein options to consumers. And ultimately, we can also modify maturity and be able to take advanced genetics and high-yielding crops from one region to another. So on the left side, you can see a couple of our first products we want to bring out. The first we've talked about publicly for the last year now. It's a specialty corn hybrid that I'll talk about in a little bit more in a moment. The nice thing is it shows very clearly a simple application of this technology. It's a foundational product for our future development in that it's a deletion that we've made by just snipping out the gene responsible for the waxy phenotype. It's a simple deletion by doing 2 cuts. And we anticipate bringing this to the market by the end of the current decade, a product, as you can imagine, since it's about a 5-year window, that we started just less than 2 years ago. You can see also an example of a disease-resistant trait product. Northern corn leaf blight is an important pathogen of corn. responsible for pretty large losses around the world. Certainly in North America alone, it's worth more than $1.5 billion on an annual average basis. In this case, we can do what I said earlier, which is to swap a gene that does not condition resistance with one that conditions good resistance from a more exotic variety, you might say. And we're in the process of working on that. We think that can come to the market within the first half of the next decade. So, to illustrate a little bit more what we've done and the concept behind the use of the technology, let me just talk for a moment about Waxy corn. So Waxy is a specialty starch corn product. It's a small acre product, less than 1 million acres a year out of the 90 or so million acres in the U.S. It's been known for a long time. Waxy was discovered over 100 years ago. There are a number of different variants of Waxy that have been well known. The market's familiar with it. That's been very attractive. It's identity preserved, which is also very attractive because we can easily manage the launch of an initial product in a very controlled way. And we received, in discussions we've had with a range of stakeholders, some input about the kind of products that might make sense to bring out, and the fact that we can identity preserve this was very appealing to some of those stakeholders we talked to. We also know that because we don't breed directly for Waxy, we breed for our large markets, and then we introduce that Waxy variant, into our elite hybrids. What we end up with is a variety that doesn't perform as well as the elite hybrids themselves. The genetics are not as good as they could be, and we can fix that by being able to go directly into our advanced inbreds. In each case, delete out the Waxy gene and be able to create hybrids that are identical in their performance to the elite hybrids we sell that are non-Waxy, and do this in a very efficient fashion. And we've already done that editing, shown this works well in the greenhouse, and this year we're testing this in the field at a number of locations. We've already worked through a process with the USDA, and they've confirmed that the next generation Waxy corn is not regulated by the U.S. Biotech Regulatory Services as a GMO. So this is a product we can bring out in the U.S. in a very efficient manner. The next slide just gives you a little flavor for some of the uses. I'm not going to spend much time on it, but Waxy is something we're very familiar with. It's in some specialty food uses. It's in industrial uses. I'm sure all of you have used paper products that have a shiny surface that take advantage of the specialty starch characteristics of the Waxy starch itself. So it's a type of product that the supply chain is very familiar with, which makes it a very appealing first product to bring forward and get our customer base very familiar with, as well as the ultimate end user to become familiar as well. So we believe that genome editing, CRISPR-Cas for targeted breeding has a tremendous amount of value, not just in our markets, not just in the crops we work in and the farmers we serve, but in a wider range of crops and customers. And I want to spend a couple of slides just sharing some thoughts about that. We believe that we have we take a sort of responsibility, I would say, for enabling others to benefit from some of the technology that we're developing. We're positioning ourselves to be a first mover with this technology. We've in-licensed technology. We've invested significantly in patenting different ways to apply the technology. And of course, as you can see, we're working diligently to bring products forward. We've already started to collaborate with other groups. We're working in a very open innovation type model. And the first example of this collaboration approach is illustrated on this slide 14 in an alliance that we've struck with CIMMYT, a public-private partnership. It's an umbrella alliance we structured, and the first project under that alliance relationship is to address a really important disease that's occurring, as you can see in the lower left, in East and South Africa, in the ESCA regions of Africa, called maize lethal necrosis disease. It's a viral disease, which means there's no chemical treatment that can help. And even if there were, most of the growers that CIMMYT serves couldn't get access to that. And you can see on the right that the level of damage can be 30% in moist environments, and this is a really serious disease for smallholder growers. CIMMYT's already working on addressing this by selecting for genetics with improved resistance, but we know that over time, as we identify those genes in partnership with CIMMYT that are responsible for resistance, we can begin to deliver those resistance genes in a more efficient manner to new germplasm as the disease might spread. So we think this is a great example of the power of the technology, taking advantage of naturally occurring variation for resistance and being able to bring that variation into elite varieties. Of course, in the produce industry, we know there are many, many applications that could be interesting. You can see some pictures here that show crops where we can see interesting applications. There are many more, and we can talk about that in the end in the Q&A if you're interested. But everything from disease resistance in spinach to reduce chemical treatments, perhaps in a longer horizon, addressing problems like citrus greening. We think there's some near-term, attractive opportunities, and we're talking to, a number of other people and companies who will address these types of markets to help enable that to happen through IP we have access to and technology we've been developing, given that there are a number of requirements to make this technology actually be effectively applied in a given crop. Now, as we think about this technology, as we've started to think about this in the last couple of years, we've recognized that there's always a challenge in bringing a new technology to the market. And that's true whether it's a technology in food or biology broadly or in any other sphere. And so we have taken very seriously trying to do this in a way in which we work closely with the society at large, all elements of the society at large. We know that achieving social license will be critical to realizing value for this technology, not just for us, but for our customers and for many, many other segments of agriculture. So we're engaging with a range of stakeholders. You might think of traditional stakeholders, supply chain people, as well as non-traditional. We're certainly talking to NGOs, around the world, environmental NGOs and agricultural NGOs, former regulators, trying to really understand how they see the technology. We're listening to consumers today, how they feel about technology and food broadly, what's important to them, so we can help make sure that this technology has achieved social license and we can see value from it in the future. We know that that technology adoption is critically dependent on achieving trust with the consumer, with our customers, and we're working hard to deliver that trust, really to earn that trust by engaging with people, by having conversations, in fact, like this one today, and others like it. So we remain hopeful that we will see a good outcome here. We've established our own set of guiding principles to help us and our entire organization think about how to use this technology wisely. And some of those examples include that when we talk about CRISPR-Cas, which is a tool that can be used for different outcomes, we're really talking about CRISPR-Cas for advanced plant breeding. And in that context, we're talking only about working with genetic material from the target plant or target crop. And so the simple way to talk about that is it's corn genes in corn all the time. It will be tomato genes in tomato only. And basically in the natural locations that they can be found that you could deliver through typical breeding systems. We're also committed to open and transparent communications about the technology, about what we're doing, about the products we're developing. And of course, we're working with regulators around the world to support appropriate science-based regulatory oversight for the products, the plant products and the crop products from CRISPR-Cas targeted breeding. So as I come to the end, just a sort of personal reflection. I shared with you earlier, the fact that this is based on a very natural process, and natural variation, I think we're all aware, if you're working with plants, is it's abundant. And this picture just shows a woman looking out across a sea of canola, and it's all completely yellow. And I do a lot of backpacking, and I've lived in California a long time, and I go backpacking every year in the Point Reyes area along the shore in Northern California. And I've been camping there for 25 years, and I can look out every year like that woman has, and across the sea, in this case, of wild mustard. And every year it's been just plain yellow. And this past year I was out in that same campsite, and you can see in this picture, if you look a little closely toward the right, some white mustard plants, white flowering mustard plants, that I'm certain represent mutations derived from that yellow flowering wild mustard. And there are parts of the field where you can't see anything white. Just in a small segment you can see it. And so it's just a reminder that there's abundant natural variation around us, that genome editing, targeted breeding takes advantage of that natural variation that is there, that channels it, that targets it, and helps us bring products to serve our customers' needs efficiently and in a high-quality manner. So I think I'll end there, Bob, and turn it back over to you for a Q&A section.
Well, thank you very much, Neil. That was extremely interesting. We're getting a number of different questions coming in, but I'd like to remind you all that if you do have a question, just type it in the chat box area at the bottom left of the window, and then just click send. So I'll get started with some of the questions that have been coming in. And you know, I was wondering, Neil, if you could expand a little bit. I was intrigued by that concept of kind of an open innovation model, where you're seeking out partners to, I guess, kind of bring some of the technology that you've been developing at Pioneer to further advance its use and to meet some of the challenges that they might be facing. It's kind of a unique model. Can you share a little bit more about that?
Sure. So, there's many different ways to think about open innovation, but, in essence, it's about how we want to collaborate and enable others with this technology, and how we want to work with collaborators in a broader sense, beyond just genome editing. But in this example, we've developed tools and technologies that we think could enable others to more quickly be able to create edits, to create new alleles or harvest natural existing alleles. We know that not everyone is set up well to do that efficiently, so we're working and are open to conversations with small companies, certainly large companies, academics who want to develop products for testing, and in some cases, depending upon the crop, perhaps to actually work with them to create edited alleles that they can then test. In some crops where we don't work, perhaps just helping them with maybe through IP relationships or licenses or technology transfer. We think there's a tremendous opportunity here. We're talking to other groups, other international ag organizations, entrepreneurs, and would be very interested in hearing from folks in your organization or elsewhere about their interests and finding ways to help them be able to realize the promise of the technology. So we continue to evolve how we're doing that, but I think later this year we'll have more clear and public ways of doing that. Hopefully in a simple and framework that makes this not too complex to navigate for people.
All right, very good. Thank you. A question that comes from the listeners here on the webinar is, there's a number of different questions that are kind of along the same lines. Are there any regulatory conditions for CRISPR at the moment? USDA had an announcement earlier, I guess it was last year, on some work that had been done in their laboratories in mushroom, but what have they said about this from a regulatory standpoint? And I guess a similar question would also apply to elsewhere around the world, in particular the European Union.
Right. So the regulatory environment, it's early days. We know that international regulations are complex. The USDA has shown willingness to be a leader in thinking about how to regulate products coming through this technology. One of the steps that currently exists is that the USDA welcomes people to send a letter in and say, okay, here's the product we're going to develop, here's how we developed it. how do you think it should be regulated? And when you send in a letter, they will give you an official response to tell you how that product would be treated. So the mushroom example you mentioned earlier, the USDA responded to their letter at about the same time they responded to our letter about the waxy corn, and they got the same response, which was that that product would not be regulated under the sort of biotech regulatory system, and therefore would be treated as a conventional conventional food product. So, we know in the U.S. that you have the USDA. There's also the FDA that has oversight over food. In general, the FDA wants to understand if you're developing is available if one is developing a new product to consult about that product, and we've done that with them. So I think there's a number of simple steps that exist today. The weight of evidence so far for the types of products we talked about, deletions, these kinds of swaps, suggests that the USDA is on track to treat those as to see them treated as conventionally bred materials. The USDA is also going through a process of reviewing its own regulations, and again, they've taken some leadership here, I think, and considering how products should be treated, and are again looking at a very, very low threshold, an appropriate threshold, we think, in terms of the safety of these products, that if there are these kinds of simple edits and deletions, that in the main they would be treated as non-regulated materials.
Very good. Another question that's, I'm going to paraphrase a couple of questions that have come in, but it kind of speaks to the one of the last slides that you were putting up, Neil, and it really is in recognition of some of the controversy over what have become known as GMOs over the last few years, and just mistakes made or not made, depending on your point of view in terms of communication. But you talked about your desire and the work that you're doing to be very transparent, engage a lot of different groups. Could you give us a little more background on that and how you're really hoping to develop the right communication framework around this technology?
Yeah. So we're giving a lot of thought to how to communicate, what the right language is, and also I think part of in a way, let me just rephrase or maybe make sure I understand where we want to go here. Part of the question I think you're asking, Bob, is how can we do something differently than happened maybe with GMOs to make sure this technology is accepted?
A much better question. Yeah, very good.
That's where it seems like.
Yeah, that's where I'm going. Yeah.
Okay. So, we've thought a lot about this. We challenge ourselves with that question. We've been challenged by others internally, and we're trying to be thoughtful. And one of the things I think that we know is that we need to be transparent. I think when you and I were first in this business, when I was a young scientist in the 80s, I think approaching this from the science standpoint, thinking this is pretty simple, why wouldn't anyone like this technology, is a somewhat naive approach. And we recognize that when people think about their food and the fact that they don't control their, the food supply, they're going to look at that and want to feel comfortable and trust the people who are developing those new products. And so we are reaching out, we're communicating transparently. We've thought a lot about the language to use, the frame of reference. For this audience, we talked a lot about breeding. For your average consumer, I'm not sure breeding is a very helpful construct today. Mm-hmm. Most people don't know where their food came from, why there's a different variety of apple than there was 3 years ago, where Braca flour comes from, and things like that. So we're still learning, and very open to engaging with folks on this phone who have thoughts about how we can do a good job in communicating. Sure. I think trust is the foundation, and I think with that in mind, we also know that by enabling many different people to use it, not only the technology, not only is that good for the industry at large, but it also may be good for overall adoption of the technology. There are many people who maybe have already developed trust in the industry. Maybe small companies have a different relationship to consumers. Maybe the International Ag organizations do. And so we feel that the more diverse are the users of the technology, and those therefore speaking about the technology and its benefits, and they want to emphasize, and its benefits in a tangible way, the more likely this technology is to be accepted, adopted, and we think that would be quite different from what actually happened with what's called GMOs today. As Bob and I well know, we worked on a range of not commodity crops, of everything from lettuce to grapes and tomatoes, but I think the ability of that technology to deliver on solutions that the consumer really wants was not so simple, and I think there's a much, I think, an easier path to that, and I think that will help in overall adoption and acceptance of products of targeted breeding.
It's actually a great transition, because I'm getting a number of questions here from attendees that are asking about, how amenable are fruit and vegetable varieties to using genome editing. So a lot of the work you described is in kind of more traditional row crops, but how do you see this being, deployed in fruits and vegetables?
So, first of all, I have many friends still in academics and other parts of the ag industry, and I know lots of people are looking at and already developing edited, varieties of a wide range of crops. I just came back from a meeting in Amsterdam a couple weeks ago, and there were lots of discussions about and presentations about applications in everything from grapes to apples to lettuce to peppers, tomatoes, of course. So I think if you step back and think about what is it we need to know and be able to do to make genome editing possible, you need three things. You need, first of all, knowledge of the genomes you're going to target. You can't edit a text if you don't know the text. And so you need to have not just a reference genome for the organism, for the crop, but you need to know the actual sequence of the crop you want to target, of the variety you want to target. Now, 10 years ago, that would have been an impossible task, but we're getting to a point now where that's a pretty accessible task. So the good news is that in terms of genome knowledge, it's going to become pretty tractable going forward, not just for a reference genome in the lab, in an academic lab, but for commercial varieties. Secondly, one needs to be able to do editing, ideally directly in the varieties of interest. And again, we've made a lot of progress as an industry in figuring out how to be able to introduce these editing systems, these CRISPR systems, into a wide range of varieties and be able to get edited alleles back out, edited varieties back out efficiently. So that's definitely a skill, and not every group has that, but it's accessible. And then finally, it's just the technology itself, the ability to design the right guides that will work efficiently, that will minimize, cutting elsewhere in the genome and maximize cutting where you want. That's another skill that, again, is going to get, I think, more and more accessible over the next years. So all in all, I know there are many crops that have been sequenced and many different varieties sequenced. The solanaceous crops, the cucurbits not far behind, some of the tree fruits. And I think being able to introduce molecules into some of those are getting also pretty accessible. So I do think we're going to find it quite possible to be able to edit alleles or harvest natural variation in a wide range of produce crops and address some really important issues that I think and that consumers would like to see addressed and the farmers would like to see addressed. Everything from improved disease resistance in those crops to maybe better tasting, higher quality produce at a nice price point.
So is the freedom here that you're getting? I mean, I remember back when people were first putting genes into plants, and we were looking at all the grand things that might be doable. We talked about changing flavor, or the texture of foods and things like that, but found very quickly that these are multi-genic traits. So is the real value of gene editing, Neil, that first of all, we have better genetic tools today, and we can come up with these genome sequences. But secondly, you can actually go in and make multiple changes to change different characteristics of multigenic traits?
I think that's exactly right, Bob. So I think it's two things. First of all, we can make multiple changes at one time, which would have been vastly more difficult, right, in the past with other technologies, and even with breeding. Trying to combine multiple, changes in a pathway, let's say, into one variety would be very challenging. I think the other thing is also thinking about, the ways we get there. So I mentioned tomato, and so for me, I think about, you have this tomato that's easy to produce, and but it doesn't taste very good, and you want to think about how do you make it taste better. Well, that's probably pretty complicated. But you also have heirloom tomatoes, which taste great, but they're pretty expensive to produce. It may be, in my view, that we've learned how to make tomatoes more producible. And maybe those are pretty well-defined characteristics. Maybe that has to do with, harvestability in some fashion. Maybe that has to do with a gene for ripening. Maybe it has to do for a gene, shelf life. Maybe a gene for how the fruit stays on the plant. And we can introduce those traits into the high-value varieties rather than doing it the reverse way. And I think that can be pretty accessible, again, with maybe 3 or 4 traits that impact producibility. I gave the example of maturity as an interesting one. You might think and say, look, we've got a great set of varieties for this maturity region in soybean, but not in this other region. How do we make this other region's varieties more higher yielding? Or maybe you address it and say, well, how do we simply move maturity by 3 or 4 genes that impact maturity and bring that great yielding genetics to a different region? So I think it's both what you said. We can do multiple changes at a time, but also perhaps rethinking the paradigm of what we do and what we've learned from all the genetics over the past 20 years in the range of crops that many people focus on.
Neil, knowing the comprehensive approach that you guys are taking, I was wondering maybe if you could comment a bit about how this technology fits in with sustainable agriculture. It's a very important topic in our industry right now, and sustaining our businesses, protecting the environment, and doing the right thing. How does this technology fit there? I mean, there are a number of different traits I would think that you could target that would result in better uses of resources and things of that nature.
Absolutely. We think a lot also about sustainable, production systems. We certainly think that issues, for example, like reducing environmental load of chemicals, right, or environmental load of other inputs is one of the key pieces of more sustainable practices. So is better water use efficiency. Now, some of those are easier to think about addressing than others. And of course, we also know that sustainability has to do with just fundamentally improved yield. So, if one is using a practice that requires, I don't know whether, there's a lot of debate about organic versus conventional and what the yields are and this sort of thing, but if one can just drive up yield, that is, and I think this can help accelerate that and give more, better realized yield, then actually you can, minimize the environmental impact just in terms of habitat. It's probably more of an issue for large acreage crops, obviously, but I think that's part of the story. I certainly think disease resistance that can be introduced and can minimize, say, fungicide applications is a real issue. And take something like banana, which is sprayed, as you know, all the time, or grapes that are sprayed heavily. I think being able to introduce genetic disease resistance that's durable by combining multiple resistance genes through this technology is a very tangible benefit that I think is very realizable. And I want to be careful not to suggest an overpromise about what's possible. It's the limitations of our knowledge of the biology of the traits we care about, but there's a lot known about resistance to a range of diseases. And a good example is powdery mildew, which is resistance to powdery mildew. In barley led to the identification of MLO genes. Those genes exist in all plants. A deletion in that MLO gene can give rise to resistance in almost any crop to mildew, to powdery mildew, and I think that's an issue in a range of produce crops. So I think that's a very near-term tangible outcome that has significant sustainability benefits. I think drought tolerance is a little more complex, but perhaps there are opportunities there as well to make incremental changes. And I think it just comes down to case by case, looking at for a given crop. And I don't know all the crops, what the real issues are in, in the produce industry, but, what are the critical issues that contribute to sustainability, and we can interrogate, is there enough known about the genetics to deal with that? But I think disease is a great area where, major impact can be made, and it'll be an interesting issue to think about how the organic industry, other types of industries, other types of practices, think about adopting the technology with benefits like that could reduce chemical load, whether it's a natural chemical or a synthetic chemical.
With that, Neil, I'm afraid we're going to have to call this one to an end because we want to make sure everybody gets done on time. But again, for those of you there's a couple of questions that we didn't get to. We have them recorded, and we'll try to get the answers back out to you via email. And I think I'd like to, again, thank you all for joining us on today's webinar. I'd also like to extend a special thank you to Neil and the folks at Pioneer for participating today. This is a complex area, and I can tell you that I heard Neil speak a few months back, and it was one of the most concise and clear explanations of how CRISPR is used that I'd heard, and so thought it would be a really good fit into our webinar program. If you have any additional questions or comments for our speakers, please use the contact information you see on the screen, or you can email me at bwhitaker@pma.com, and we'll certainly try to get you the answers. To learn more about issues affecting the industry and technologies that can help solve them, you can join us at PMA's Technology Symposium. It's going to be in Monterey, California, May 4th through 5th, and you can learn more about that by visiting pma.com/events/technology. This webinar was recorded and will be available on pma.com in a couple of weeks. This concludes today's webinar. Thank you again for your participation.